Good morning, everyone. I'm Matt Reynolds. I'm filling in for Radha today. I'm the Associate Chair for Research and Entrepreneurship in Electrical Engineering, and it's a pleasure to welcome our speaker today, Ellie Shlizerman. Ellie is an assistant professor in the Department of Electrical Engineering with a joint appointment in applied mathematics. He received his PhD in applied math and computer science from the Weizmann Institute of Science, then spent 3 years as a postdoctoral researcher at Princeton, and then also in UW Applied Math, and was promoted to assistant professor very recently. Ellie's research focuses on classification and modeling of dynamics of complex systems, for which he develops methods that combine data analysis and dynamical systems theory with real data, which I think we'll see today. He collaborates with the biology department here at UW, UMass Neurobiology, and the Allen Institute for Brain Science. Ellie has received the Boeing Research Award and the Joint NSF-NIGMS Initiative Award at the interface of mathematical and biological sciences. His work on the olfactory system was recently published in Science and covered by the New York Times, BBC, and others. So with that, thank you, Ellie, for coming to speak with us, and let's welcome our speaker.
Thanks a lot for the very kind introduction. It's a pleasure to speak at UWEE Colloquium. And today I would like to give an overview talk of my research. My research is combining methods from dynamical systems and data-driven discovery to study realistic networks of neurons. In particular, our focus is on understanding these structural properties and dynamical properties of these systems. What we would like to learn from these systems is how do they process data, and we focus on sensory systems for this purpose. We also would like to look at the architecture of these systems and see how we can mimic this architecture or to understand better how these systems are designed. We also look at the adaptation of these systems over time, and with the intention of learning how they learn and how do they adapt to the environment and the stimuli that they are being exposed. The research, computational research in neuronal networks is being more and more possible in the recent years due to technological advances that allow us to really look into these systems. One important advancement is the ability to map these systems. And in this respect, the Connectome project, which is mapping the neural system architecture for different systems. There is the human Connectome project. There are other connectomes that are being established for neural networks. And the ability of these technological methods is to take the neuromorphology, the anatomical structure, as you can see here, and to transform it to a graphical representation, to a graph where nodes are neurons, and the connection between these nodes, the edges, are the connection between these neurons. This is a very useful structure for us in terms of computational methods to start to study them and to see how they are designed. But this is only the beginning of the story, and I would like to give an example on the C. elegans network. C. elegans is a worm, and it's a fundamental model in biology in general. In neurobiology, this model is its connectome was fully resolved. It has about 300 neurons, and we know all the connections between those neurons. In particular, as you can see here, there are two types of connections in this network. There are electric connections, called gap connections, and there are synaptic connections, which are chemical connections. And by visualizing the system, you can start seeing the differences between those connectomes. Those graphs are really different. One is the real small world network. Another one looks like a very dense graph. Also, with this visualization, we can start seeing which neurons we expect to play a significant role. Those are marked with large radiuses here. They correspond to the nodes that are hub nodes in this network. The edges between each pair of nodes here correspond to the number of connections between the neurons. And also, we can classify them into different types and so on. So indeed, when this connectome was resolved in the middle of '80s, it was a lot of excitement in the field of computational graph models is to resolve what is actually, what are the building properties, and what can we understand from looking at this graph in terms of the capabilities of the C. elegans worm. But over several years, with different attempts of community detection, of detecting different motifs in this graph, it seems like this is only part of the information that we have about the neural network. And the another part of the information is actually that this is only one layer of the network. The additional layer of the network is the dynamics of it. So actually, this graph is dynamic. Each node in this graph is a neuron, so it's integrating inputs that are coming into it in time. And also, the edges are dynamic and correspond to the different interactions. As we saw, the connectome already has two types of connections, and these are very different types of connections in terms of their dynamics. Okay? So combining these two layers together is producing for us an actual model or an actual system that is able to perform. And we cannot do with only one layer. So when we add the dynamics to the connectome, we are eventually getting the dynome of this network. And this is something that we would like to study. So for the C. elegans worm, we have developed a dynome, and I would like to show it for this network. This is a project with an undergraduate student, Jimin Kim. And in this network, so what we are showing here is the connectome, the synaptic connectome of C. elegans. And first, you can browse through the different neurons, see how they are connected to each other. Some neurons are, like this one, are hub neurons. Some neurons are much less connected, like this one, but have very strong connections to a particular other neuron, and so on. But what would be really useful is to now play with this network. So we allow this possibility. If you pick a neuron here, or you go in the list and pick a neuron, particular neuron, so I'll pick a pair of neurons to excite and hit the start button, eventually what I will see is the voltage, which is dynamically plotted on top of the connectome. And what you can see is actually that neurons are suddenly coming into play, are starting to have particular dynamics here. Some neurons are idle. Many neurons are idle. Many neurons are really responding to this input. And with this dynome, we can now inject inputs into any subset of neurons here and to watch what kind of dynamics we will get. So this is really a great capability for us. So with the dynome, we went to a C. elegans worm network and asked, can we reproduce or at least visualize the neural dynamics that correspond to different behaviors. One of the behaviors is locomotion forward. And you can see the worm is now crawling forward. And experimentally, it is known what are the neurons that are supposed to promote this locomotion. These are actually those neurons that I've picked in the dynome simulation. So when we will inject current into them, and we will look at the time lapse of the voltage dynamics of these different neurons, we will see that this group of neurons is starting to be active. Some of the voltages are negative, some of the voltages are positive, some of the voltages are oscillating. If we will look at it for a long enough time, we will see that there is actually a periodic pattern here. So those excitations persist in a periodic fashion. And also what we will notice is that our intuition from a graphical graph theory point of view is completely wrong here. So we identify that these neurons will probably play an important role in the network, but in such dynamics, they are just idle in this excitation. When looking at another initiation of the network, and this excitation of LML neuron will correspond to, so LML neuron here will correspond to backward movement, what we will see is the following time lapse. So notice what is different here is that different other subsets of neurons are being active now. We can see also a different period, different amplitudes of these neurons. So the hope of to find one neuron that is associated to a particular movement, or that we will reveal all the information from the connectome, is just not working for those realistic systems. So if we will go back to this illustration that was shown in Scientific American to explain and illustrate the human connectome project, and asks how do we untangle the wiring of the brain. It was published in 2010, 5 years ago. How will we revise it to nowadays? I would say that we will add a power supply here. So we will power the network, expose it to stimuli, and then we will ask the questions of, if we really want to untangle what the wiring of a particular neural circuit is doing, we have to dynamically simulate it, and then to conclude what this network is doing. But the main problem with that is that these networks are extremely complex, right? Only the C. elegans connectome has more than 7,000 connections, 300 nodes, and it's almost impossible with some intuitive methods or even analytical methods to study those systems. So our proposal is to look at the data that is collected from those systems, to place sensors at particular neurons and also edges, and then to record this data, and from this data to conclude what are the possible dynamics, what are the capabilities of these networks, and what can be concluded about them. In some systems, we can record data from the actual system, so to apply multi-neuron recording techniques. I've brought with me sensors that we are using to record, so I will pass them around. What you'll see here, those are multi-neuron recording electrodes of 16 channels that are being used to record from the olfactory system in moths that I will talk about. And also a very small electrode is used to record from mosquitoes. Okay. So when we gather this data, there are several aspects that we would like to answer in those systems. The particular aspects are, first of all, when we don't know what the system is doing, what we would like to do is to classify its dynamics. Then we would like to ask the question of how the system is being designed. What is the connectome of the system if we are not familiar with the particular design of the system? And the last part is, what can we do with it? How can we synthesize more dynamics, as I showed with the dynome? And how can we apply control techniques and learning techniques to this network. Okay, so let me start with classification because it's really the fundamental step where we gain understanding of the system. The classification task is more like a clustering/classification task, where we sample the dynamics of the network, and then we would like to divide it into subclasses, clusters that are separable, and this would be the training step. And then the next step would be we would like to get a new sample of the dynamics, and then to recognize to which subclass it belongs. Okay. And here I gave an example of C. elegans. So these are the classes that we kind of want to map, like locomotion, sensing, detection, turns, and so on. So there are several levels for the classification. When we talk about gathering data from sensors, we can apply, of course, feature extraction techniques. But we have to remember that this data is from multiple sensors, so it has some spatial structure, and also depending on time. So it's a multivariate set for different samples as well. So applying feature extraction techniques usually will not result with low-dimensional representation and with only few clusters or few classes that we are expecting. A natural data structure that we can think of for those recordings is a matrix where we really separate between the two dimensions that we have. So the rows dimensions will be the neurons dimension, and the columns dimension will be the time dimension. We call this matrix, so this is an actual time lapse of the system that we sampled. And we called each of those matrices a snapshot matrix. And then the idea is to transform this matrix into a low-dimensional representation with spatial patterns. Those are the red neural codes. And when we project those samples, those snapshot matrices onto this basis, we will get low-dimensional dynamics with the hope that we can analyze those low-dimensional dynamics, separate them from each other, and assign them to a particular class. So we are working on methods to develop such tools. First, so I will describe a bit about the methods that we develop. But first, I would like to also emphasize what are the difficulties in working with such data sets. Well, the first difficulty is that we have a spatiotemporal structure. So this matrix has time dimension and space dimension, which are the neurons. Since it's a matrix, first, it's a natural thing to apply matrix factorization techniques to see what are the dimensions in this matrix. Like, for example, singular value decomposition that will decompose the matrix into spatial modes, time modes, and the singular values will tell us how many dominant spatial modes we have. Many times, this procedure will not return the most optimal low-dimensional representation because of the spatiotemporal structure. But more than that, we are actually collecting a whole collection of these snapshots, and we would like to obtain a joint optimal basis. And then we would like also to obtain such a basis that will be as easy interpretable as possible, because we would like to analyze those eventually trajectories that are projected to the space. Okay, so just to show, so the example that I will use to show how we construct a classification space is classification of scents. In general, in a sense, it's a really interesting problem. When we smell coffee, we immediately recognize it as coffee, but it has many chemicals in it. It's composed into many volatiles that are in the air, and only a particular composition of them corresponds to this particular scent. So we'd like to understand how these independent chemicals are combined together to produce a particular neural perception. It's a collaboration with the UW Biology lab on olfaction with the Riffel lab. So what we do, we record from insects, which olfactory sense is extremely sensitive and corresponds to behavior. And the idea is to break a particular scent, the flower scent, into basic chemicals. This approach is also used in other neural systems. For example, in the auditory system, in speech, we can break words into letters or sounds, and then to see whether we are able, from these independent responses, to combine them into one response that makes sense, into a sequence. So we break the flower odor here in the example to 3 different chemicals, and then record from multiple neurons, about 100 neurons in the network. And here is an example of firing rates, which is sliding a window and counting how many spikes each neuron spiked for this stimulus over time. And you can see that those matrices are nowhere simple to analyze. And we don't only have one matrix, we actually have a collection of those matrices. So for each chemical, we are collecting those snapshot matrices. And then our goal is to produce a combined joint classification space. So just to show that a direct approach won't work, I've combined those snapshot matrices into 3 combined sub-matrices, and then applied singular value decomposition, showing that 95% of the energy will be represented by about 50 modes out of 100 modes possible. So it means that the reduction is not that successful. Only for 3 chemicals, I'm expecting to get a space which is 3-dimensional. I'm getting a 50-dimensional space that I need to consider. And if I will project onto the space, I will get trajectories that are very tangled together and eventually don't provide to me a good separation of these independent chemicals responses. Another approach could be separating those snapshot matrices into independent matrices and applying matrix factorization techniques onto each matrix separately. This approach actually works and produces only 1 or 2 modes per each submatrix. But the problem is now, how do we combine those patterns into one set? Because it's really important for us, because if we will take, for example, here I'm taking each dominant mode from each factorization, combining it into a library matrix, and then projecting the snapshot matrices to this matrix, and obtaining that the trajectories projected to this basis are inseparable. Okay? So although there is an indication that I grasped the right structure here, the right pattern here, I'm unable to distinguish between the trajectories onto them. So one of the methods that we introduced is really simple and kind of combining clustering with factorization or dimensionality reduction techniques. And this method goes to this library matrix that we obtained from separate applications of factorization, and asking which, for each neuron, so each row here in this matrix, supposed to show on my computer. So each row here is corresponds to a neuron. It chooses from the responses the maximal value and then zeroizes all the rest. It kind of associates a neuron with its response to a particular stimulus. And this is what is depicted here. So eventually, for each neuron in this 3-dimensional space that I would like to construct, I will associate it to one of the axes according to the maximum value, and will zeroize all the rest. And it turns out that this method works really incredibly well. So here are the same projections, but now onto this new space after the exclusive threshold reduction. And now you can see that the trajectories are very well separated from each other. And also we can start analyzing their dynamical properties. So each trajectory goes to a fixed point in the orthogonal space here, and really recognizes this particular stimulus as that orthogonal direction. And that's the key construction in the exclusive threshold reduction, how to construct an orthogonal basis out of non-orthogonal vectors. More than that, now we can use the classification space to consider all those mixtures and see how do they correspond in the representation. So here is a mixture where we took an actual scent and mapped it to the classification space. And now, according to this area of the fixed point of the trajectory, we can characterize what this mixture means in terms of mixing those particular chemicals. I won't go into the details, but we actually enhanced those methods by asking what would be the most optimal basis to consider, and can we reweight maybe those responses such that we will get optimality in this representation. And also then we considered how one can think of singular value decomposition in an iterative way, such that we already incorporate those dynamical properties inside the matrix factorization technique. Those methods are based on the idea of enhancing the norm in singular value decomposition such that it will be compatible for optimality concerns. Some of algorithms not for time-dependent dynamics were actually introduced by faculty here, like Mariam Faisal and so on. So we are inspired by those methods and took them to spatiotemporal collections of responses. So this is the result of eventually construction of the classification space, is that we can recognize stimuli. So we constructed a classification space for two scents here, verbena flower versus the female moth, and now projected different trajectories onto it, also identifying the decision regions that correspond to the fixed point that corresponds to each stimulus. So here, for example, when stimulus flower is given to this network, the projection will result with horizontal trajectory that eventually will enter the decision region of the flower and will indicate that this particular stimuli was applied. And this process can be repeated and repeated, eventually get a very good distribution of the actual stimulus that is being applied. Here you can see that the other stimulus will produce vertical trajectories that go into the other decision region. When there is no stimulus or other stimulus being applied, we will not get any correlation with the decision regions. So as I mentioned, insects are extremely interesting to study those responses because it's really their behavioral sense that they rely on in order to survive. The Datura flower is a resource for the Manduca sexta moth, which is a nocturnal moth, and it finds this flower in very dynamic conditions. So what the experiments, the behavioral experiments that are done in Riffel lab here are simulating this dynamic environment in a wind tunnel, where the wind blows from right to left, and with the wind, the particular scent is being released, and it looks something like that. It's a turbulent flow, and then they track the trajectory of the moth attempting to reach the source of the scent. And those recordings, as you can see here, this is an example of a really successful trajectory. The moth used only a bit of time in order to identify the source of the scent, and then immediately cruised towards the source of the scent. So in an aim to pollinate the flower. And what has been identified is that if there are backgrounds that are being added to the scent, or the frequency of puffing the scent is being changed, then the moth could be confused and sometimes even ignore the scent completely. Eventually, those are behavioral measurements that, that we were able to obtain, saying that the preferred frequency of puffing is 1 Hertz, otherwise it's being confused. And also some backgrounds won't interfere with the neural with the behavior, but some background scents will mask even this behavior of finding the flower. So we did, in parallel, we also analyzed the neural responses, so recording from many neurons, and then analyzing of how many recognition do we have for particular scents. And we found the following results, as you can see here. And the main message is that we actually found an incredible comparison between, or correlation between our results, which are the neural representation results, and the behavioral results. This was an aha moment for us, where we said, okay, so if we have such an incredible correlation between neural representation and behavior, let's also check what happens to chemicals that are really loosely related to the Datura flower, but correspond to pollutants and some chemicals that could be in the environment. And we indeed investigated that. And yeah, that's the cover from Science journal. We published this work eventually in Science. And what we concluded is that some chemicals completely mask the behavior and the neural representation of the olfactory network in moths. And some don't interfere with it at all. Okay, so it's first evidence that those environmental chemicals can influence the representation, and also a way how you could recognize and classify neural responses. Continuing with that, so this is a work in progress and a future work. We are also thinking of now, if you have too many stimuli, then representing it in a graphical topological way could be too complicated. So we are developing statistical methods to represent the classification space as a probabilistic graphical model. And here is an example. The goal in these models is eventually to ask queries about particular stimuli or particular initiations of the model. Okay, once we establish the classification space, we can now discuss about the structure of the network. So we already understand some classification, some separation of the dynamics. Can we reconstruct how the network is being designed? So with the olfactory network, we exactly addressed this question, and we asked if we have a dynamical model, which is an implicit model, we don't know how the neurons are connected. We just have motifs of connections or particular connections that we know whether they are positive or negative. And we obtain enough samples. Can we reconstruct the connectivity that will support such dynamics? So our novel viewpoint at those inverse problems is to combine the classification space plus model reduction to make sure that the reduced model will produce the dynamics that we are looking for. So particular system that we apply to the solution of inverse problem is looking at the antennal lobe. This is the system that we have recorded from in moths, and which processes stimuli. olfactory stimuli. And as you can see here, it's really interesting what is the role of those internal neurons, the purple neurons that are called interneurons. They don't get, how the processing of olfactory stimuli works is that there are receptor neurons, those neurons, and there are many of them. Then they project their input into projection neurons, which are output neurons, and those are the neurons that we have recorded from. But they also project to interneurons that back then connect to projection neurons and modify their response. So it's really interesting to understand how those connections are designed such that the response in projection neurons will be coherent or correct. Okay? So we identify those connections, inhibitory connections, from interneurons to projection neurons as the connections that we would like to find. And wrote an implicit model for this network. And here's the implicit model for each population of neurons. And the matrix B is the connectivity matrix that we would like to infer. And as I mentioned, the goal of the approach here is then to project this system onto a reduced model, and then to match this reduced model in an analytic way, actually in procedural way, structural way, with the classification space that we expect, and then to infer the connectivity. I won't go over all the steps and technical many steps of the reduction approach, but we'll just show a picture that explains it. We are planning such that we'll have an orthogonal fixed point for each input in this system. And then this results eventually in an optimization problem for the connectivity. Once we solve this optimization problem that is convex in this case, we are getting the connections and can wire the network such that it will perform the dynamics that we asked it to perform. In particular, we can even prove theorems. So once this optimization problem is solved, we can prove that this network will perform what we expect it to perform. And this is a really great benefit from the approach. It's not a fitting approach that we can just think of something and hope to get the result. We can actually prove that this optimization problem will produce the correct network. So we applied it for an example. Here you can see for 2 inputs what would be the calibrated matrix. We also explored how it will respond to very noisy stimuli, which are the environment around the olfactory environment around insects. And what you can see here is that the advantage of this network is that it's much better performing for enhancing the signal to noise ratio versus random calibrations or no calibrations at all. Okay. We also constructed it for several sets of real data, like you can see here for 107 neurons, for 2 cents, 3 cents, up to 9 cents. We can reconstruct this connectivity matrix. And eventually, one of the examples looks something like that we get a full connectome of the network. And this network is supposed to produce this particular functionality that we expected to produce. I'm showing this picture because I think it's cool. But also another reason is that when looking at this picture, you wouldn't guess that it is producing 2 particular cents, and discriminating all other cents. So it really looks random, but actually it's calibrated. It's designed to produce particular dynamics. We are now working with this kind of a connectome. We would like to implement it on a microchip such that one could take this network to the environment and sense from multiple sensors. And also in in a spatial environment, when you introduce several such sensors, how do you combine data such that you will be able to sample a whole spatial, a whole region, and then to combine it into eventually a classification method for identifying particular rare events that need attention and so on. Another example for the success of recovering the structure of the network is navigation in monarch butterflies. So monarch butterflies are really a miracle of nature. They fly more than 2,000 miles during their migration period from northeastern USA to central Mexico. So, and they do it using a time-compensated compass. So they do it in an optimal way, such that they know how to identify the azimuth of the sun, and according to that, to adjust their orientation, flight orientation, such that they will always fly in the right direction. So the main question is, how is this mechanism implemented within the monarch's brain? So the eyes of the monarch are, here is a demonstration of the eyes, and you can see when the sun moves, indeed, it corresponds to a different projection on top of the monarch eyes. So that would be the azimuth signal. And then it is combined with several signals that encode the clock. And the question is how this integration mechanism inside the brain is combining them into a compass. So it's a very difficult question because you need to look everywhere in the monarch's brain in order to identify and then also to find the actual analysis. So we approached this problem by using dynamical system tools and also saying, what should a control mechanism have in the classification space? So if we are going to plan such an integration mechanism, what should we include? And we concluded that in the azimuth time of day plane, we should have a line attractor. So a line where all points on this line correspond to an attractor such that any initial condition will eventually converge to the fixed point. Now going from that, we can construct a Lyapunov function that describes convergence to this line attractor. And then we can integrate the inputs that could be recorded, those azimuth encoding inputs and clock inputs, to ask whether we have such a mechanism. And it turns out that for these inputs, we investigated many possibilities, so 128 different configurations of the network. And we concluded that only one configuration of the network will correspond to the right stable flight orientation over the whole course of the day. You can see this orientation. And those are the signals that we are recording from the clock and the azimuth inputs. So this is really interesting how dynamical systems can help to navigate to the right solution with combining with data. So eventually, with this compass mechanism, we can guarantee that the butterfly, for any time of day, notice here, this is the morning, and the butterfly started at about 40 degrees, eventually will converge to the right orientation. Here is another scenario where the butterfly starts at 240 degrees and eventually converges to the southwest direction, but it follows a much longer path to converge to it. And this is a property of the model. What we found is that except of the stable fixed point, you also need to have an unstable fixed point, a separatrix. Otherwise, this mechanism cannot be implemented. And this means here that the butterfly cannot cross the separatrix and have to go much longer radial way in order to converge to the southwest. And those conjectures eventually were being confirmed using flight tracks recorded in a flight simulator outside in the environment. And we see very similar features, like the existence of separatrix doing several circles until it eases into the right direction, and so on. We are planning now on recording from neurons that we suspect that do the integration and verify the mechanism. So with having these two components, both classification and connectomics, we actually have a network that we can play with. So we can start thinking about how we can really investigate whether the network is capable to produce coherent movements like this one, or it's incapable, or what should be added, or what kind of control should we introduce in order to support such a movement, and so on. So I would like to go back to the C. elegans example and to show how do we model it and apply different control techniques. So first of all, when looking at many neurons in the C. elegans network and recording them, so this is a snapshot matrix of motor response voltages, what we can see is that we have only two modes that correspond to those movements. And you can see the singular values of those modes. All other singular values are almost zero. And when we will project onto the space of these two modes, and we will look at time, we will see that this is really a periodic trajectory. So those simulations that I showed in the beginning of the dynome are really corresponding to periodic trajectory and could be represented really optimally in two-dimensional space. So this is a forward movement. Then we looked at the different neurons and asked, OK, so do they really correspond to moving the body when we will position them along the body with different amplitudes? And this video shows where we move the neurons according to their voltages is that we really get coherent patterns in the body, not only in the neural dynamics, but they persist in moving the body or moving the muscles in the correct way. So this suggested that we can then go from neural system to connect it to the muscles. The muscle map is also known. And then to connect those muscles to a mechanistic model, we model it as a viscoelastic rod that will simulate the worm moving. Okay? So we wanted to test whether it will work. So here is the simulation of the neural system controlling the elastic rod through the muscles. So about 1,000 of ordinary differential equations are being integrated here to produce this movement. And we are exciting only this pair of neurons that is supposed to be associated with forward movement. So I'll play this video. So we start with some initial state. And what we see is that really the body is forming into this coherent movement, eventually corresponding to forward crawling. Since the excitation is constant, and we already know that we have a limit cycle, this movement will proceed forever until we stop exciting those particular neurons. So one of the suggestions was to use it as a screensaver. But we also examined then backward exciting neurons and asked, will they produce a movement that moves backward? And this is this simulation. As you can see, indeed, the worm starts to move backward. The movement is more jittery than forward. And indeed, we associated it with, it's not really a preferable movement of the worm. It usually likes to move forward. So it's kind of degenerate case. But the direction is really clear backward. We then asked, is it really the case that all excitations will kind of produce coherent movement? So we did small perturbations of the excitation that we used for forward and backward movement, and we are eventually able to produce only those kind of movements. What you can see is that other forces are not producing any interesting dynamics, actually, like very atypical dynamics, non-coherent. So small perturbations will really suddenly ruin the whole behavior, and we will not get the desired movement. So I would like to finish the talk by saying that with having the model, we can actually think of control mechanisms as well. So here, once we have a model of the neurons, and it's connected to a viscoelastic rod model, then we can ask, OK, so what should be the input then into this network to promote such movement and to lock into particular behavior? So that will already be a recurrent network. And we developed a particular algorithm that will find the weights according to the movement of the worm into sensory neurons, and then to find out which sensory neurons are the key sensory neurons that will produce such behavior. We are in particular using a variation of a Kalman filter force training approach for that. And this video demonstrates those dynamics. What you can see is that a competition between constant excitation of forward exciting neurons, feedback into this neuron, and also feedback into a subcircuit of neurons. And what we can see is that for short time, this is our conclusion out of this optimization routine, is that for short time, having the feedback into particular specific neurons actually wins, right? So the magenta worm wins here. But over time, we need to find the exact feedback. Otherwise, it will just stop, and eventually the movement will ruin. So we can find this optimization, but only for a short time. OK, with this, I would like to summarize to mention that we went over classification, different approaches for classification. For future, we are working on multi-layered classification that will consider different aspects, different classes, and also how we can combine classification in space as well, so crowd sensing for sensors. Also related to classification, but also the structure of the network, we would like, instead of having these dynamical models, once we investigated them and ran a lot of simulations, we would like to summarize those simulations into a probabilistic model. And this is what we are working on. Also, visualization techniques are extremely important here. You saw how the visualizing the connectome and putting on top of that the dynamics really shows us what are the neurons that are playing the role and so on. And the last part is learning, implementing, and also understanding how learning and adaptive control is implemented in those systems. I would like to thank students that I mentioned involved in all those projects, my collaborators, and also you for your attention.
We have time for a couple of questions. Any questions? I have one, which is about the experiments in learning dynamics of the worm motion. Mm-hmm. Is there any sense in which one of those particular learned combinations is energy optimal? In other words, is some combination of activation of dynamics more favorable energetically, so the worm should be more efficient?
Yeah, it's a really interesting question. We cannot yet relate it to energy favorable ones, but we see that indeed when we are looking for a particular solution that will promote a movement, and we are targeting all the neurons in the network, we see that usually our algorithms will fail to converge. Only when we point at the specific neurons, then we are getting convergence. So it means that energetically favorable perhaps is is really knowing the key of the neurons and not spending the input into all the neurons and produce a movement, but actually having a subset of key neurons that are command neurons and promoting particular movement.
Cool.
In insect investigation, indeed, we are seeing that optimality in terms of energy is definitely exploited. So the existence of the attractor and using the attractor for navigation of monarch butterflies is really the optimal solution. Otherwise, they won't be able to migrate for such distances.
In your local networks, you've got motifs for that particular function, that neural subgroup. Are you finding, or have you investigated, if the density of motifs are the same as they would be throughout the entire neural network, or are they, did the motif density change per subnet?
Mm-hmm. I was asked to repeat the question, so I will repeat the question. Is the density of motifs is global or particular to parts of the network? That's a really interesting question. So the density of general of some connectomes is very different. So we see that the synaptic connectome is much more dense than the gap connectome. And there are several biological kind of reasonings for that. When we go inside the connectome, we can look at also densities within this connectome with particular neurons, and whether this indicates something about the dynamics. That's very difficult to say. So graph theoretical research was really focused on this question for a pretty long time, and it wasn't able to correlate between particular properties of connections to particular properties of functionalities. One of the conclusions is that after 3 passes through the edges, you will actually cover the whole connectome. So there must be something additional to it that incorporates the dynamics. And this is really the dynamical rules, how the neurons are able to interact and to suppress some connections and promote some other connections, even if they are not related directly to the density of the connections.
Great. Well, thank you very much, Ellie, for speaking with us.
Thanks.